Melissa. Our next speaker is Dr. Lisa Keith. Now, Lisa could not be here in person, but she's pre-recorded her talk. And one of her lead technicians, Blaine, is here for the question and answer session at the end, okay? And while they're bringing up Lisa's talk, good. Lisa is a research plant pathologist at PBARC. She uses a combination of lab, greenhouse, and field-based techniques to develop early detection tools for emerging pathogens and deliver practical disease management approaches that can be adopted by growers. And again, Lisa couldn't be here in person. And at the end, Blaine will be able to answer any questions that you might have about Lisa's research. Okay? Will you be able to?
Hi, I'm Lisa, and I'm a research plant pathologist with Ag Research Service in Hilo. And the topic of my talk today is managing coffee leaf rust with fungicides. And I figure I can give an overview of all of the work that we've been doing with fungicide testing, that being in the laboratory or in vitro testing on potted plants and also in the field, and then end with a summary. This is our disclaimer for the mention of any product or trademark when it comes to working at the USDA. So before I actually get started with the work, I want to just mention the incredible team that we have, not only in my lab, but at our center, working collaboratively with university, with SHAC. Also, we would not be able to do what we do as researchers without the growers and the producers. So we thank you so much for your collaboration. So just to remind everybody again, I know you've heard this a million times, but the disease coffee leaf rust, the pathogen Himalaya vistatrix, essentially spores start infections that develop into spots to produce more spores. And it's critical to have an understanding of this pathogen. The spores will land and actually germinate and enter on the underside of the leaf. And then they tap into the leaf tissue with these specialized structures called haustoria, which are like feeding structures. So they don't kill the tissue right away. They actually feed off it for quite some time and produce mycelium within the leaf. And then germinate back out to produce more spores on the underside of the leaf. And that is what you're seeing, that powdery yellow spore production. And those are a picture of why it is difficult to control this pathogen, just this sticky nature of these spores that can resist all kinds of environmental conditions. And the disease does vary from season to season. It really depends on rainfall, not only for germination, but for the disease cycle. So to begin with lab fungicide testing, these are what we call our in vitro tests. The simple methodology is collect fresh spores, actually mix them with the product in a tube, let them sit, plate them on a water auger, which just looks like clear jello on a plate. Let the spores incubate at 22 degrees, somewhat optimum for the fungus, in the dark for 12 to 15 hours, and then look at the spores under the microscope and actually count the number of the spores that have germinated as compared to a water control. So this definitely looks like a busy slide with lots of information, and it is. We've looked at probably, 25, 30 plus products. What this graph is representing is on the x-axis, each individual product. And on the y-axis, it's the percent germination of spores. And so again, it's the mixture of the spore with the product, plating, and now determining if being in contact with the product inhibits spore germination and how much. And so I'll point out that towards the right side, the control, so that's just mixing the spores with water. On average, for the close to 30 times collecting spores at various times of the year at various locations, the average germination rate is approximately 55%. Okay. But the range, we've seen it as low as 32% germination rate and as high as 72% germination rate. So now when you compare that control to the various products, you can see that there are varied effects on germination of spores. Now, I tried to color code it to make it a little easier to visualize. So the yellow highlighted area are the systemic fungicides and Preaxor. Okay, the light blue highlighted area are the copper products. So things like Badge and Cueva and Coside. Anything with the green highlight, I grouped into biological products. Now, that can be bacteria like Serenade or double nickel. That I grouped in any kind of essential oil product, natural product. We get a lot of calls to take a look at various products to see if it will, in fact, inhibit spore germination. The last, just to kind of preempt for the field studies, we wanted to see if a spreader or sticker did anything on its own, and that's that light brown. So as you're looking at this graph, you can see that there are varied results. There are products that pretty much inhibit all spore germination, and then there are probably a group of products that contribute to approximately a 50% reduction in germination rate. And then there are others at the right side where there's virtually no effect on spore germination. Now, I want to, I guess, kind of say that it definitely, you have to read the label. You have to understand what the active ingredient is. You have to understand the mode of action of these products. And this is really only taking a look at if indirect contact, the product affects spore germination. Now, some of the products are, you see effects once they're on a plant. So of course, something like that will not actually affect the spores. But overall, you can see that the systemics, of course, work wonders at inhibiting germination. I'll point out that Preaxor at the couple rates, low and high rate, did quite well. The coppers, of course, are proven as far as for inhibiting spore germination. Some of the biological products that are labeled for use, like Serenade and double nickel, definitely affect spore germination. And then there are lots of things that we've looked at that have no effect. So the in vitro tests, it is not a be-all and end-all of testing. Okay, it helps us prioritize what we look at in the field. It helps the work we do to help support the university and the IR4 program. But this is just to show you that a variety of results, depending on product type, can directly inhibit germination. Okay, so this moves on then to what we did with some potted plants. And in this case, we took healthy seedlings, we applied a treatment. In this case, we compared Serenade with Badge and a water control. So all of the potted plants got sprayed with the product dried, and then the plants were inoculated with spores on day 0, day 7, and day 14. We observed for symptoms, taking average ratings once a week for at least 4 weeks, just to see, well, because these are protectants, if they're there, how long does it offer protection? And again, this may not exactly equate to what is happening in the field, but it gives a good clue. Okay, so for controls, this is 4 weeks post-inoculation. Really, anywhere 3 to 4 weeks, we start to see those chlorotic infection spots on the upper surface and already massive spore production, again, on the underside of the leaves. Okay, when we look at some of the pictures taken with badge or serenade, now you can see with some of the product and the residue kind of gives some clues. This also is 4 weeks post-inoculation. In this case, some of the badge at the bottom with the blue, you can see spore production, and we'll get into that and explain maybe why we saw things like that. Okay, so here's another graph, and we tried to summarize the overall data. The x-axis is showing weeks post-inoculation. So again, all plants were sprayed, as you can see with the labels on the right, and then the plants were inoculated with spores either the same day, 1 week, or 2 weeks post-spray. And on the y-axis is the average rating, which were the counting either of the lesions or essentially looking at severity of the entire potted plant, okay, the diseased area of the leaves. So for example, the scale of 1 is less than 1% severity, and you can see 9 being more than 75% of the leaf covered with spores. Now, if we look at the water, blue, orange, and gray, that followed the typical progression of disease symptomology. Within 3 or 4 weeks, you start to see the spores, which gradually increase over time. Okay, when we look at badge, the dotted blue and the orange and the green. Now you'll find that badge 1 week and 2 weeks inoculation. So again, the potted plants were treated and then inoculated a week or 2 weeks later. There was excellent control over the time, and essentially there were no symptoms that developed out to 7 weeks post-inoculation. Interestingly, when badge was applied to the potted plants, allowed to dry, and immediately inoculated, we saw minimal control, and we hope to explain this a little later. For serenade, the same day inoculation with red, and the 1 week post-inoculation brown actually showed excellent coverage, but coming now 14 days. So treating the plant and 14 days later, we saw that there was not as much control, which actually matches to what the label use is recommended, approximately every 14 days. Okay, so this moves into the field testing portion, and I'll readdress what we thought happened with badge towards the end. But so for 2021, we had a field trial, and the results showed that the rotation of badge, serenade, and double nickel would allow for really good control of CLR in the field. So we use that as our control in the 2022 field trial. And overall, in the farm in Holualoa, there were 13 treatments, 1 being no spray at all, 1 being water and a surfactant. Then there was the control rotation badge serenade double nickel applied with a mist blower and a backpack sprayer. And then the comparison of ProBlad, a combination of Instill serenade and double nickel. There was Cafidac, Priaxor, Alto, Approach, Apruvia, and Excalia, again, to help support the IR4 program with the systemics. Okay, so this picture actually shows the field trial separated into 3 blocks. And so each treatment was repped in 3 blocks, approximately ranging in elevation, probably about 500 feet different from the top block to the lower block. Okay. Essentially, the test started week 0 in May, okay, with the first application date of May 26th for all products being sprayed, and a second application about 8 to 9 weeks later. Now, this shows you that into week 13, week 15, you're now into September. And for the rating scale, we looked at incidence, so the percent of leaves infected with CLR. And I'll mention that overall, especially when the test got started, incidence was extremely low. So we started taking data, but actually amended the incidence ratings to include the entire tree. So instead of just picking a few branches, high, mid, low branches, we actually did observations on the entire tree to get ratings. Rating 0 being there's no rust. A rating of 1 is less than 5%, and you can see 2, 5 to 10, and it goes up from there. Okay, so this is one shot in time in August, just to show you the average whole tree incidence rating and the comparison of all 3, the average of all 3 blocks. Okay, so again, you have the 4 controls being either a no treatment, water with the widespread, or the rotation of badge serenade and double nickel, and then the comparison with ProBlad, with Priaxor, and with the systemics. And at this stage, I want to point out, even though it looks like yes, there are differences. If you note on the scale, at this stage, late August, there was very low incidence. Okay, so the entire field was less than 5%. Okay, when we look again to see, so the previous slide was the average of all 3 blocks. We were interested in seeing if there were differences between blocks. And so red, green, and blue shows you blocks 1, 2, and 3 with the various products. And there definitely appeared to be a few areas that had hot spots. In block 1 overall, probably because there was less airflow, closer to a house and a rock wall. But again, overall, everything remained very low incidence. Okay, so this summarizes the whole field trial. So from starting in the sprays in May, and then data taking every 2 weeks. Here it shows trying to, again, spread out the data and also spread out the average incidence on the y-axis. I've noted with the black dotted line that the incidence is less than 5%. And we kept expecting, because we would see some spots, that the treatments would start to lose efficacy over time. And that is what we saw. It's probably easier to talk in trends of what we saw. And then I tried to group that according to color. So the red the greens, and the purples. And so overall, what the field trial was showing that the systemics, which included Preaxor, showed excellent control, essentially, maybe just a couple percentage, very few spots overall. Then you had the grouping, the purple grouping up top, which definitely increased in incidence over time. But interestingly, you had this large group in between that hardly reached that 5% threshold by harvest. And I'd like to point out that that includes the controls. So not only the spray rotation of badge serenade and double nickel, which held up quite nicely, but there no spray control, and including just water and widespread. But this isn't to say, oh, that should be a treatment we can just spray widespread. That's not what we're suggesting. I guess overall, we're showing that in a field that has good fertilization, good pruning, the trees are healthy when the disease starts, and products are applied with good coverage, including Preaxor and the systemics. You start to see really good control at managing the population of spores. And because the systemics and coppers helped remain the spores kind of in check, this allowed for even a no spray control that was within that mix to have very low incidence for quite some time. Okay, so I know that was a lot of information, and you definitely can reach out to me later or ask Blaine questions. But I wanted to go over again why it's important to get good spray coverage and to understand how these various products are move throughout the leaves, throughout the plant, and what happens when the fungicide is applied. Okay. So you can see the blue droplets are being sprayed. The fungicide's being sprayed onto the plants. And the first plant you see is the contact, the protectants. So we're talking badge, serenade, double nickel. Spots land, droplets spread minimally. Okay. They don't travel. They don't become systemic. They just land on whatever surface it is to offer some protection. Okay. The middle plant is, well, the translaminar, which definitely spreads throughout a leaf. It can go from one leaf surface, from the upper surface to the lower leaf surface, but it does not protect future leaves, future growth. It does not go systemic, like the picture on the right. So the systemics, the true systemics, which are being tested and hopefully label approved through the IR4 program, it will travel through the vascular system. It will help protect the new growth. It will help good coverage wherever that may land on the pre-existing leaves. Okay. And this is, again, to show you that picture. Well, why are protectants? They need to be on the surface really before spores land. And yes, the protectants like copper, we've seen even badge and serenade knock out spores, actually inhibit spores, decrease percent germination. But if they're not already present on the leaf, it's hard to offer more protection. Okay, the true systemics in the middle, they're within and throughout leaf material, which also travel to the new leaves. So when spores land and germinate inside leaf tissue, those systemics will knock out spore production and actually kill the fungal mycelium growing within the leaf tissue. Okay. Translaminar, like Preaxor, will go from one leaf surface to the other, so the upper to the lower. Again, the spores are germinating through the lower leaf surface. So a translaminar will actually get to where it needs to be for that particular leaf. Okay, so good spray coverage is key. I want to kind of touch back to why maybe we didn't see the expected results for badge in our potted plant assays. Okay, so you see symptoms on leaves. The left being those chlorotic spots. Well, that's the upper surface of the leaf. So if you were to apply a protectant at this stage, it will not actually reach any of the fungus, okay? And you're not actually hitting any of the spores. The right picture is the lower surface of the leaf, those orange spores, any of the biologicals and copper-based products. The protectants need to reach this surface to not only affect what might land, but to help knock out the spores that you're observing here in this picture. Okay, so this was our spray the copper product on the potted plants, let it dry, and immediately inoculate. And we were surprised to see spots and spores develop over time. But when you zoom in to some of these pictures, this is a good example and a good lesson for why coverage is key. You can see kind of an uneven coverage, and there was no sticker spreader with this. So you see where the droplets landed. They did spread a little, but the spores obviously hit areas close to stomates that weren't already protected, which allowed them to germinate inside, actually get established, grow back out, and produce abundant spores. Okay, so I'll just give a 1-second update on a couple other projects that we're working on. One of our favorite projects, looking for natural enemies of CLR and our overall mycoparasite survey. We've been surveying now a variety of locations for close to 2 years. We're up to close to 200 isolates. That's the picture showing the diversity, representing more than 42 genera. And we are now doing assays to see, that's what the bottom right picture is showing you, that when you have a bunch of spores, as the top left is showing you, and you add these mycoparasites, what do they do? You can see they start to colonize, and we're measuring how they're affecting the spores overall. Okay, we're also have a lot of variety testing going on, ramping that up incredibly, working with leaf discs in the laboratory, which allows us to test a large number of varieties over time. And just to show you at the bottom right, the comparison of susceptible, okay, a lot of spore production. We've found some tolerant or resistant where you see chlorotic spots, but there is little to no spore production. And we've also, I'm happy to say, that we found some resistant material, which actually doesn't even allow for visible chlorotic spots to be produced. So more information to come in future talks. So I'll just end with this. Overall, and I know you've heard this collectively from researchers, and the message really hasn't changed since the discovery of coffee leaf rust here in Hawaii. It has to be managed continually. There is no silver bullet. You have to use the integrated pest management approach with short and long-term strategies, okay? With Melissa mentioning a number of these as well, you have to scout regularly. It can be tricky, especially at the early stages of these chlorotic spots. It's best to improve and maintain plant and overall soil health, having good hygiene and sanitation in the field, working in proper pruning, to use all important tools, including these fungicides, the protectants, Preaxor, and hopefully soon some of the systemics. Ongoing research, hopefully we'll find some good and good tasting resistant varieties. and the prospects of actually using biocontrol, some of these mycoparasites, to help manage as part of the program. Again, we want to avoid the introduction of new races. We currently only have one, race 24, and we want to avoid evolution of new races. So just because there will hopefully be some resistant varieties doesn't mean you can stop all management practices. And with that, I'll say thank you very much. Again, please reach out. There's my email if you have questions. I thank our funding source. I highly suggest you check out Andrea's website for the abundant amount of information there. And I thank you very much for your time, and take care. Thank you.
Okay, you can see that our scientists have been real busy for the last 2 years. We've learned a lot. A lot of these fungicide trials were, single product treatments to see, how long maybe we could, what's effective, and how much time, we can spread out the sprays. But in the next year, the plan is to start looking at sequential treatments. So a series, it may not just be one product. How do we rotate products, okay?